Thank you all for being patient. I left an extra hour to get here from Seattle, but of course there were 6 accidents and that only helped a bit. So my name is Anne. I work at the University of Washington in the Department of Genome Sciences. I've worked in several labs, but for the last 20 years worked with fruit flies, and I've come to really, really love them, and it's hard to imagine studying anything else. So today I'm going to be talking about lessons from the fruit bowl, what fruit flies teach us about human biology. And like most scientists, I'm used to talking to other scientists, but I'm happy to be here. And I didn't really know who I would be talking to, but I assumed there might be a few film nerds in the audience. And have any of you seen the movie The Fly? Okay, so maybe half of you or so. I won't be giving away too much here, but for film nerds, people might be interested in interpreting a movie according to its dramatic structure: the exposition, the rising action, the climax, falling action, and then, of course, the resolution. So I thought in describing my research and the research of my colleagues that I would also try to follow this story arc. And we're going to be starting off, let's see, for our exposition, I'll tell you a bit about the beginning of fly genetics. And then I'm going to go into 3 mini stories or mini plots of examples of how fruit fly research has illuminated human biology and human medicine. For rising action, we have a story about fly development and what we learn about human development. The climax will be an exciting story about tumors in flies. And that'll be followed both by some falling action, an interesting story about fly sleep. I don't think that will put you to sleep, though. And then a resolution that is a little bit about the future of fly genetics. So first of all, I'll tell you about the beginning of fly genetics. So when I talk to ordinary humans about flies, they understand that a lot of scientists are interested in fruit flies and that it makes a convenient model organism, partly because of its very fast life cycle. And in fact, flies do have a nice, quick life cycle where from the time that a pair of adult flies mates, they lay an egg. Oops, I'll get my pointer here. A fly will lay an egg. That egg will hatch into a larva, which goes through 3 molts before it pupates into a pupa, well, like its little cocoon. And then it comes out as an adult, which can then mate and repeat the cycle within 10 days. Contrary to a lot of, preconceptions that flies then just live for a few hours, a fruit fly adult can live for about 1 month and maybe even longer if it's kept well fed and so forth. One single fly can have hundreds of offsprings. And so if you're a scientist where you're interested in getting a lot of data, that's a great way to get a lot of data quickly if your single fly can give you lots and lots of babies. The cost of maintaining them is very low. In the laboratory environment, we raise the flies in these plastic or glass bottles, which are corked with styrofoam, and each bottle has a layer of solid food. It could really be made of anything, but we use like molasses and cornmeal. And you can see that each bottle has just hundreds and hundreds of little flies in it. So the pioneer of fruit fly genetics is Thomas Hunt Morgan. And in the early 1900s, he started growing flies in the lab. And just sorting through natural populations of flies, one day he happened upon a fly that had perfectly white eyes and everything else looked normal. So like any geneticist, he of course mated that fly to another fly. In this case, he took the male white-eyed fly and mated it to a regular female because that's what was around. And within 1 generation, that male, I don't know if you can see this, had thousands of progeny. But all of the progeny had red eyes, male and female progeny. But then he took these progeny that had red eyes and mated them to each other. And now he started to see really interesting patterns emerge where I won't get into the nitty gritty of like how, this gene works. But after the second generation, he had easily 6,000 flies. I think he probably had more, but he probably stopped counting because he had, solid results here. Finally, in the second generation, he did get white-eyed flies back, but they were all males. And from these experiments, he concluded, well, first of all, that eye color is hereditary, but it has this thing where it skips a generation as a recessive trait. And more interestingly, because this sort of thing was already known from other types of genetic studies, that this trait was sex-linked on the X chromosome. And this was the very first time that anyone had been able to prove the chromosomal theory of heredity. And for this, he won the Nobel Prize. So I just want to point out that although the fruit fly wasn't maybe the necessary animal to do these experiments with, it may be one of the only animals with which you could get so many data points, almost 10,000 data points within a month's worth of research. One of the advantages of studying fruit flies is that they basically all look identical. I mean, aside from like the occasional white-eyed fly that pops up in a natural population, they all are very, very stereotyped with the same number of bristles, the same number of stripes, the same size, and so forth. But along the way, Morgan's students started to not only look for natural rare variation in the populations, but also intentionally mutagenize the flies with X-rays or with chemicals. And they started to pull out different physical traits, like different eye colors, some of which are recessive and some of which are dominant, and different eye shapes, some of which are shown here. Here I'm just showing examples of the eye, but, again, they accumulated hundreds and thousands of different physical types of aberrations that affected the wings shape and the, body color and so forth and so on. So that's all cool. Like I'm interested in this stuff, and maybe some of you think it's cool, but really, is this research relevant? Especially to people who fund science. Is the research relevant? Most people think, is this relevant to human biology and human medicine? After all, humans are sophisticated machines. Even the simplest of humans can usually see, hear, smell, taste, and feel, learn, digest, metabolize, poop, make babies, grow, defend from predators, heal wounds and infections, navigate through the world. These are all complex processes that take, coordination between different organs. But nevertheless, all of these sophisticated processes are also things that flies can do. Flies, you could argue, are also sophisticated machines. And one idea behind all of this is that through evolution, animals have had to figure out these things. And as soon as an animal figured it out, and as evolution proceeds, we don't reinvent the wheel. So the way that a fly is able to metabolize food, even though its internal organs look different than a human, on a cellular level, is really, really quite similar. Genetically, flies can do all of those things with a much simpler genome. A fly has just 4 pairs of chromosomes compared to a human 23 pairs. On those chromosomes, they've got 13,000 genes packed in there compared to the human 20,000. So not that much difference in genes, really, as far as gene number. There is, though, an 18-fold difference in the amount of DNA material. And it's thought that the human genome has, in some cases, duplications of genes, or also information outside of the genes that gives maybe a fine-tuning of when and where that gene is turned on or makes a protein. But nevertheless, we can learn a lot about these genes in the fly. Well, we can learn a lot about human genes from studying the fly, because in fact, at least 75% of genes that are in the human are also found in flies, and many of these genes are also disease-causing genes. So flies have been a successful model organism to study a lot of human diseases, and I'm listing just some of them here: congenital disorders, cancer, sleep disorders, addiction and alcoholism, congenital disorders. Sorry, I have that twice. It's very important. Parkinson's and Huntington's and other neurodegenerative diseases, infertility, hearing disorders, aging, obesity and diabetes, infectious disease, and tissue regeneration. So I could probably go on, but for this talk today, I'm just going to focus on 3 of these stories, the top 3 here. And so bringing us back to the outline. The first part of the plot, I'm going to tell you about fly development and what we've learned about this in flies that we can apply to human biology. So in the fly, the body plan is established very early. Here we're looking at a fly embryo just 5 hours after fertilization, and already there's tens of thousands of cells. And if you stain this embryo with fluorescent antibodies to look at what genes are turned on where, you can see this stripe pattern. Where actually here's the anterior of the embryo, and then it kind of wraps around to this posterior part. And these genes are already, even just 5 hours into development, planning the anterior-posterior body plan that will become the adult. So it's telling the embryo, you're going to be the head and you're going to be the tail. And interestingly, this wouldn't have to necessarily be the case, but it was discovered that these 8 genes, actually 7 of them are shown here, but there are 8 genes, they're all aligned right next to each other on the same chromosome in order from anterior to posterior. And so, for example, I'm kind of turning things around here a bit, but this green region here, this green gene is telling the fly, okay, you're going to make this segment, and you're going to make a halt here, which is a very tiny wing, which you can't see quite in this picture. I'll show up the next animation. It's the green, dark green would be this haltier here. It's like thought to, there's like these little mini wings that act like gyroscopes to the flying fly. And then just anterior to that is another gene that says, okay, you're going to make the segment that makes the wing. And so thus it makes a wing here. However, along the way, there was, when scientists were x-raying flies in the early 1900s, they came across a mutant in which they observed that instead of this forewing and little mini hind wing, they ended up with 2 identical wings. And this, it turned out, was caused by a genetic aberration that made essentially 2 copies of that wing gene there. Now what's cool, now this is cool because now you have a cool fly with 4 wings. I think that's cool. But more importantly, we have found out that the wheel hasn't been reinvented, that this idea of the body plan and the genes that are spatially arranged on the chromosome is something that's conserved across every single animal. So for example, in the developing human embryo, it has these same exact genes arranged the exact same way. In the case of the human, though, it just gets a little more complicated where this situation is set up on 4 different chromosomes and, some duplications, but also some little bits of refinements here and there, basically. And interestingly, in humans, it's been shown that, like in the fly, loss and gain of these different genes leads to many human congenital defects. And Ed Lewis did a lot of this work. He's starting in flies and then bringing it into mice and humans, and he won the Nobel Prize to acknowledge the importance of this work. Another example of how fly genetics has led to insights into human development and congenital defects is the example of the gene eyeless in flies, which is a gene that turns on very early in development that says, okay, you are going to make the eye. It turns on in the right time, in the right place to make an eye. So whereas a wild-type fly would look like this, a fly that's missing the gene would miss its eyes, therefore the name of the gene, eyeless. And if you do the opposite experiment, where instead of taking away the gene, but you add more of it back in to kind of pump up the volume, the fly ends up with extra eyes in places where it normally wouldn't have eyes. An extreme example of this is where you can force this gene to turn on in the developing legs and end up with flies with eyes where it should have legs. Now, it turns out that the reason I'm telling you this is that humans and flies both have the same gene, essentially. It has a different name in humans, but if you do an alignment between the DNA of the human gene and the fly gene, there's tremendous similarity on the DNA level, but once it's turned into a protein, there's enough similarity in the DNA that the protein itself ends up being 100% identical. And as proof of concept of this, if you start off with a fly mutant that doesn't have eyes, you can genetically take that mutant and patch back in a copy of the fly gene and then rescue that defect. Moreover, if you take the fly mutant and add in a copy of the gene that originally came from a mouse, it also rescues that same defect. So it's just showing that those proteins are functionally completely identical, even though they come from animals that are spaced through, many years of evolutionary difference. Now, as I say, that wheel of how eyes develop really hasn't been reinvented. In humans or mice or flies that have a functional copy of this gene, all these animals have normal eyes. But where the gene is defective, it leads to severe defects, not only in the fly that I've already shown you, but in mice, and in humans where there are many cases of people that are born with a mutation in this gene, and it leads to severe blindness. So I've told you about how we can learn about human development from flies. Now I'm going to switch topics for a second and tell you about an even more extreme case of studying tumors in flies. So one mutant that was discovered by happenstance along the way is a fly that, instead of looking like this, like a nice normal fly, starts to grow horrible tumors all over its body. It's amazing that this fly even made it, because most of the animals with this type of mutation would die much earlier in development. So this won't make much sense to you, but this is a piece of the developing fly larva, the wing disc, which doesn't look much like a wing yet. It's inside of the larva's body, but it's going to be programmed later on in development to make the wing. In any case, it has always a very stereotyped pattern and size, but in fly larvae that have this same mutation, many of the wing discs end up super overgrown and looking like this, so this animal is just going to definitely die. This gene that was mutated was named hippo after the phenotype because of these big tissue foldings and just gross abnormalities, although the hippo might be offended. But it turns out, as you would predict maybe, that this gene, and why I'm telling you about it, is that it's also conserved throughout evolution all the way up to humans. So in mice or in humans, this gene is just called MST1. It has a second copy of it, MST2. This is a normal mouse liver, and in a case where this gene is mutated, the liver is super overgrown. It turns out that the gene hippo, which I've told you about, is in this pathway overall that is a master regulator of proliferation and apoptosis or cell death, and the differentiation of cells, and just overall homeostasis, which means the ability of organs to kind of know what size they should be and not grow too big or too small. The gene and this whole entire pathway is conserved throughout evolution up to humans. So this is showing the mouse pathway, which has many of the same genes, just with different names and slight differences here and there. And importantly, it's been discovered that in humans, there are many examples where mutations in this pathway lead to many cancers: breast cancer, colon cancer, pancreatic cancer. And so, again, the wheel hasn't been reinvented. And it's an example where by studying these pathways in flies, we can do experiments more quickly and easily and maybe discover drugs that could interact to prevent cancer or help cure cancer. For the next mini story here, I'm going to tell you a bit about a very recent story about what we've learned in flies about sleep and human sleep disorders. So flies do sleep, even though they don't have eyelids. A sleeping fly looks just like this, which is about how an awake fly looks. But if you're watching them move in a bottle or whatever out in the environment, you would know that a sleeping fly is just standing still. So this is kind of shown here, a control fly, showing its activity levels plotted over 6 days, where here's day 1, day 2, day 3, etc. The nighttime, or when the lights are off in the lab, is shown in these gray bars, and then a white bar is daytime, and then, daytime, nighttime, daytime, nighttime. Actually, so maybe that's 12 days. Anyway, whatever, it doesn't matter. But you can see in any case that when the light is on and you're tracking the activity level of these flies, which can be seen here with these black lines, that the flies are active during the day and not active at night. And furthermore, that there's a peak of activity in the morning. They're kind of napping during the middle of the day, and then there's a peak of activity at night. Now, it turns out that it's been known for a while that the circadian rhythm of all animals is under genetic control. And so one example of a gene that is a master regulator of the circadian rhythm is the gene CRY1, where if you take a fly in which it has a mutant copy of CRY1 and plot out its activity level, at first glance it doesn't look terribly different from the control, but there are some important differences. First of all, during the nighttime, whereas a control fly is completely inactive, here you do see little mini spikelets of activity. And then a little more subtle, but also quite reproducible, is that in these mutant flies, their peak activity, they have 2 peaks of activity during the day, but it's offset by about 3 hours. So I don't know, if you imagine that this is 8 o'clock at night when these flies are having an activity level, here it looks just before it really gets dark out. So maybe here the peak activity level would be at 10 or 11 o'clock at night. So CRY1, it turns out, is in a genetic pathway in flies in which it's directly responsive to light. So light hits a cell. I actually don't know the details of this. I was just thinking that I should look it up. But this gene and the protein CRY1 is right at the cell membrane and is directly responsive to this light. When it's hit by light, it goes into the cell nucleus and then interacts with a few other genes which act in a negative feedback loop, which kind of like a loop, it turns itself on and off so that even without necessarily any more light feedback into the system, the cell is going to know about the 24-hour circadian rhythm. So again, the wheel hasn't been reinvented. If you study this pathway also in the mouse, which has been done more recently based on what we know about fly research, these same exact genes are present in a pathway that's just slightly modified. And as I've shown you, whereas mutations in this gene affect fly activity and fly sleep in a human, if you mutate this gene and track using, like electrodes in the brain and stuff, and then monitor the sleep activity. In this case, the colored bars are representing different phases of sleep, like REM sleep and so forth. In a control human, the differences between sleeping and not sleeping are pretty discrete. And if you compare day 1 versus day 14, the timing of sleep is pretty consistent. So the person wakes up at the same time and goes to sleep at the same time, wakes up at the same time and goes to sleep at the same time. In comparison, a person in which this gene by chance carries a mutation, they still will wake and sleep, but the pattern is just more chaotic. And not only is there interruptions during the wake and the sleep, but also the time at which the person goes to sleep and wakes up kind of just shifts a lot from day to day. One guy who has studied this since I think, the 60s is Michael Young. He's at Columbia University. He brought this research, first starting in flies, to studying it in humans and in human pedigree. So here I'm showing you a family tree where the circles represent females and the males, the circles represent females and the squares represent males. And then here we're looking at their offspring and their offspring. And what he did is, in families that were known to kind of have a history of sleep disorders, he tracked their activity levels and the patterns of awakeness and asleepness, and also genotyped them for this CRY1 gene. So here in red, I'm showing you cases in which these patients had a defect in the CRY1 gene, where a hatched line indicates that they're heterozygous for that gene, and the solid fills indicate that they're homozygous for that gene. Now, if he plots out their sleep patterns on a clock like this, where in this case the little circles represent the middle of their sleep cycle. So like for these non-carriers, they maybe go to sleep at midnight and wake up at 8 a.m. So the middle of their sleep pattern was at about 4 a.m. in the morning. However, people that carry a mutation in this gene, whether it's homozygous or heterozygous, their sleep cycle is shifted by exactly 3 hours later. So these guys are night owls. And you can also see maybe that there's just a little more chaos, whereas in the controls, just about everyone sleeps, with a uniform pattern. Here, some carriers are sleeping in the middle of the day when you don't quite see that as often in non-carriers. So I think it's an interesting case of bringing a story of fly research into humans, particularly because we all know night owls. And I think, even I think of it so much as not something that you're born with, but something just like a character trait that maybe you pick up through habits as you go through life. But it's just super clear that these night owls, the night owls are the ones that carry mutation, and the non-night owls are the ones who don't. So just kind of makes you think about things a little bit differently. And because this work all started in the fly, and so much of what we know about the humans came from the fly, it's another good example about how the wheel of how and when we sleep hasn't been reinvented. And just last year, Michael Young won the Nobel Prize for this work, and I got to meet him. He's a really nice guy. So finally, I'll tell you a bit about the future of fly genetics. I'm going to start off here with a quote from a book that I really enjoyed, and that if you guys have been interested in what I talked about today, you might want to pick up a copy. The book is called First in Fly by Stephanie Moore. She wrote in the conclusion of the book that, through 100 years of research, fly research, it's been full of surprises and insights, and that many fly studies have ended up having an impact on biomedicine, but they did not begin with human disease foremost in mind. They were just picking up random, like, oh, this is a cool fly, it's got 4 wings instead of 2, or, it's got huge tumors all over it. These studies began with curiosity and an interest, maybe I have a typo there, a desire to answer the question, how does that work? And then the connections to human health revealed themselves later. So as I told you at the beginning of the talk, many fly genes turn out to be very important for human disease, and I've told you about 3 examples. But as fly research and some of the tools become more sophisticated, like right here I'm showing you a fly in which its eyes are lit up with a red fluorescent protein, and the muscles of the muscles that control the wings are lit up with green, and so this fly kind of glows in the dark. So we have more sophisticated tools, but there still remain, both in flies and in humans, many genes with unknown functions. And there are still many human diseases with unknown causes, and so there's still very, very much that we can learn from the fly. And for a second, I'm going to jump to my own research, which I haven't told you about yet, partly because it doesn't quite form a complete story yet, but it's a story that we're very intrigued with so far, and to see where it goes. So the lab that I work in, the lab of Celeste Berg down at University of Washington, is interested in studying fly eggs, which have a very stereotyped morphology. Where here's the egg, and which would have a developing embryo inside of it. And just to the anterior side, there are these two kind of antenna-like structures that help bring oxygen to the egg. Now, we started studying a gene called IDGF, which we became interested in partly because it is important for human biology. In humans, it's upregulated in many, many types of cancers, and it's associated with particularly cancers and tumors that start to spread and become malignant. But when we mutated this gene purposefully in flies with defined gene editing, at first we were disappointed to see that the flies overall were normal and their eggs looked normal. However, we made a surprising observation along the way, partly because of the mistake of an undergraduate, where whereas flies, the normal and the mutated flies, lay normal-looking eggs when they have a blood with a normal pH, when you intentionally or accidentally lower the pH by exposing the flies to too much carbon dioxide, which this undergrad was doing. In the case of a wild-type fly, even with low blood pH, the flies will still lay normal eggs. But now the eggs from the mutants that I've got now turn out incredibly defective. And so we think it's an interesting example of how we're bringing genetics to a new level where, in this case, the gene itself doesn't directly cause a defect, but somehow the gene is interacting with the environment. In this case, just a subtle change in the pH in the blood, where the flies now have really, really abnormal defects. So that is basically the conclusion of my talk, but I have a bonus feature, which I want to address the movie for a second, which you're about to see. So I want to correct the record, which is that there's a scene in the movie in which our heroine is screaming. The fly is looking at her, and it does this dramatic effect where it's showing through the fly's eyes and the, what is it called, the compound eye, that it's showing just multiples of what she's looking at. So this is not actually how a fly sees. Instead, all of the little components of the compound eye each detect a particular tone. They do see in color, but in the case of this black and white screenshot, it would be more like a very pixelated image. Although flies see overall things a bit more blurry, the trade-off of this in a way, or the advantage of this, is that what they give up in definition of what they're seeing, they gain in speed. So flies are able to see much more quickly than humans. Humans can process, I guess, about 60 frames per second, and flies process about 4 times that quickly. And that's one reason why they're so difficult to catch. So with that, I will close, and I'll take questions. Or we could just watch the movie. Yeah?
Here I find that we have sort of a fruit fly season.
Ah, yep.
Is that just basically temperature, or is there something else going on?
Right. That's a really good question. So he asked, is there a fruit fly season? Well, he observes that there's a fruit fly season, and he asks if it's because of, well, I'll tell you the answer. So you're onto the right thing. So flies are quite resilient to all kinds of temperatures. Like you can even, if you have a bottle of flies, you can put them in the fridge and they'll last for probably 12 or sometimes even 24 hours. You can put them in the freezer and they'll last for several hours. They'll look like they're dead, but then they're able to wake up a little while later. So basically, yeah, flies do overwinter, and they probably do better in little, insulated environments like nearby houses and things like that. During the summertime or even the springtime, when temperatures warm up, we do see more flies because their life cycle speeds up. Their life cycle is temperature dependent, and also they survive better when the weather is warm. They also, a really big part of it though, is that since flies feed on fruit and seek out fruit, there are a lot more food sources during the spring and summertime. People's composts are starting to rot more readily, and flies love that sort of thing. Yep. Hello. Yep.
The flies that you use in your lab, have they been bred and been lab flies for generations and generations, or do you ever go out?
Oh, yeah. So, right. So that's a really good question. So are the flies that we have in the lab inbred, and do we ever, instead of taking flies that have been inbred for many generations, get flies from the wild? So yeah, the flies that my lab uses are quite inbred. They're from strains really that have originated from the same labs that Morgan started in over 100 years ago. So there are cases where we think that because of that inbredness, that there might be artifacts that we see that are because of that inbredness. And so there is more increasingly an interest in fruit fly research in testing out flies from the wild and seeing how they compare to these inbred lines. And in particular, a few years ago, I worked for a short while in a lab that was interested in fly behavior, and in particular how flies navigate through the world, how they fly and how they walk based on visual cues. And we found tremendous differences between strains that were inbred and lab grown versus strains that were collected from the wild. And we did a fun field trip where we collected flies by going to a nearby fruit stand and just like caught them with a bait and then tested their flight behavior in the lab. And we did find that wild caught flies were better flyers, like more kind of stronger, more robust. Yes? Ooh, do we expect climate change to have an effect on the number of flies? I would think yes, because of the question earlier of how flies definitely do better when temperatures are in the 70s and the 80s. I think like their optimal temperature, like if you were to be growing flies in the lab and you want the most progeny per fly, you would grow the flies at about 75 or 80 degrees. And so as our global temperatures climb to more areas that have that type of temperature range, I would expect more flies, as long as there's still food to feed them, right? I mean, you can imagine if climate change causes droughts where there's a lack of produce, then there might be fewer flies. Okay. Thank you.